we're almost at the last stage. We are going to make the graph. So what we did till now was clean the data. So we imported the libraries, installed the required packages, imported the libraries, we imported the data set from Excel, and we cleaned the data, we grouped them, and we did statistical analysis. The first one was a Shapiro-Wilk normality check, and the second one was ANOVA, where we also did the 2 keys post-hoc for multiple comparisons. Now we'll compile all of this into a good looking graph, which would be publication ready. So to plot the graph, all right, so first I'm going to put a comment plotting the graph. So we're going to make a bar graph, and now we're going to be using the ggplot2 package from the tidyverse package. So in the first video we installed the tidyverse. So from this we used one package which was called dplyr. We used the mutate function from that. Now another package in tidyverse which comes along with tidyverse is ggplot2. So we're going to be using that, right? We are going to be using the compiled data frame that we named over here, ATP, and we're going to be piping this into something called summarize. We used another similar function called summary, right, for ANOVA. Although we have used summarize once when we were doing the normality check, we got all the groups. We summarized the Shapiro-Wilk test for all the columns, and we put it into different columns. So similarly, we are going to be using summarize to get the mean and the standard error of mean. We are going to keep that data available for plotting the graph, right? So in summarize, the first one that we're going to be doing is the first thing that we're going to be putting is the mean of the values. So right now we have a long data which is they're in one column. So if I hit ATP here, or I can hit view ATP and I hit command enter, you can see this is one group, this is the second group, third group, and the fourth group. So what I want is I want the mean for this. For these three I want one mean, these three I want another mean, third, and finally fourth. So summarize will do exactly that. I'm going to take out that view. Summarize is going to do exactly that. So I'm going to create a variable called mean underscore FC. So this is the mean fold change, and I'm going to put and I'm going to equate this to mean. So mean is a function in R that's going to calculate the mean, and we have to put the column name for which we're going to calculate the mean. So fold change. If we wouldn't have done the group by treatment, then what it would do, it would take all of these and it will give us one mean only. Now since these are grouped by the treatments, so we'll get one mean for every three treatments, for every three fold change. So that was the whole idea of putting the group by treatments. All right, so we're going to give a comma. We're also going to calculate the standard error of mean. So that's where the plot tricks package it comes in. So again, we're going to write sem fc underscore fc, and this would be the standard error of mean. The function for standard error of mean, it's std.error. Inside this, we're again gonna put in fold change. Next, when we are plotting the graph, we want to show the error bars. So for that, we need a maximum value and a minimum value for that for that error bar to go up and down the mean value. So normally error bars would be mean plus the standard error of mean, mean minus the standard error of mean. So I'm going to make another variable called mean fc, and I'm going to use another function called mean. That is, this is minimum. The mean was the average. min is the minimum value. So minimum value would be mean fc minus sem fc. Similarly, I'm going to put a max value, max fc, max. So max is also maximum value, it is also a function. Mean fc plus sem fc. So for minimum value, mean minus standard error of mean. For maximum value, mean plus standard error of mean. Now we are set to make the graph. For that, we're going to pipe it and put it into ggplot. Now ggplot, again, it takes the first argument as the data, and since we're using the pipe, we don't want to give that. The second argument is we need to specify what is in the x-axis and what is in the y-axis. So that x and y-axis, in case of R or in case of graph, it is considered to be the aesthetic of the graph. So we have a function called aesthetic inside it, AES, and the first value we need to go and write x equals, so our x is equals to treatment and y equals mean fc. So we want the bar, the end of the bar, to go till the mean of the fc. That is why we calculate the mean fc. Now, right now I wrote x equals to this, y equals to this. But in aesthetics, it is already implied that the first value is going to be x, so I can delete this, x equals, and the second value is going to be y, so I can delete the y equals as well. So our x is going to be treatment and y is going to be mean fc. Now, the graph in ggplot, all the graphs in ggplot, they work in layers. So till here, if I hit enter, you can see here on the right, it has put the graph without any bars, because we haven't specified whether it's going to be a bar graph or jitter plot or point graph. So it has only put the background, and it has put the treatment as well as the x-axis and the y-axis. So this is the mean fc. These all we can change later. Now you can see the \n, it is not coming. So then whatever was there on the right of \n, it goes down for 40 millimolar glucose, also for this also. Right. Now we want to put the bar on top of this layer, for all the treatments. So ggplot works by adding layers on top of one another. So we're going to hit plus. Remember, when we're using ggplot and we're using layers, we don't use the pipe because we're already in the ggplot. Now we'll just add layers. So we're going to hit the plus button every time we add a new layer. So the first thing that we want to do is put the bar graph, the bars of the graph. So for that, I'm going to write geom underscore bar. So geom refers to geometry, so how the graph will look like, whether it's going to be a point, whether it's going to be a jitter plot, whether it's going to be a box plot or a bar graph. So we're going to be using the geom bar, and the first thing that we're going to specify is the status. So stat equals identity. So we are actually saying that don't change anything, just take whatever we have piped in. Just take that and make the graph. Stat equals identity, and after that we can specify the color. We can give it a color of sky blue. I like that color. And all right, so the color actually specifies the border color. So this one we are going to give it black. The fill, it specifies the color that is inside the bars. So over here we're going to be using sky blue. All right, so it's there, and let's hit enter and see. All right, so we have a graph, we have the bars, and this is the mean value. That is right now this is the mean value. We're going to put error bars on this, and it has taken the blue color, the sky blue color, with black as the border color. So this is a bit nerve-wracking because color should be coloring the inside of the graph, but it's coloring the borders. If you don't give anything, it's going to by default go to black. So it's better that it specify the color. And I don't want it to be so dense. I want it to have a little bit of transparency. So I'm going to put alpha equals 0.5. That should work. It should be a little bit transparent. Yeah. All right, so the bar graph is set. Now we need to put the error bars. Okay, so for the error bars, again we're going to be using geom_error bar. Over here for the error bars, we need to specify the minimum and the maximum value that we set over here, min FC and max FC. So again, this works with aesthetics. So aesthetics, and the first argument would be ymean. Minimum value is equals to min FC. Otherwise, if you wouldn't have specified this over here, you can write the whole thing over here. So you can write ymean equals to mean FC minus sem_FC. So it's better to specify it at the beginning. And ymax is equals to max FC. If we do this, and if we go and hit enter, so you can see that the error bars are there, but they're looking ugly. They're spanning the whole width of the bar, and for the first bar you won't get an error bar because all the values are 1, right? So to reduce the width of the error bar, we just use a comma. Width equals 0.02 or 0.5 or 0.2, and it will reduce that. Okay, so we have the error bars, we have the ymean, the ymax, width is reduced. Next, let's tackle the labels. I don't want it to be mean_FC. So again, we're going to add a layer on top of this. So everything we're adding by putting in a plus is going on top of one another. So the first layer was the empty plot. The second layer was the bar graph. The third layer is the error bars. And the fourth layer, we're going to put the labels. So for labels, I'm going to write labs, and x equals treatment, y equals fold change, and in bracket we can write mRNA expression, right? So now if we hit enter, you can see it's fold change, mRNA expression, and treatment. And we can also put in a title. So title equals, we're using the gene ATP6, is a part of the mitochondrial metabolism, and it will come on top, right? So now what I want to do is I don't want this checkbox kind of background. I want to have it clean. So this is a part of the theme, the default theme of ggplot. Now if I want to change the theme, I have to say theme classic. There are various different themes over here. You can try them all out, but I like to go with the classic. So it's going to have a white background. You can see everything clearly. All right. So now the graph is looking a little bit better. Still, you can see we have a little bit of gap from the x-axis till starting of the bars. So I don't like that. You might like that, but I don't like that. I want to I want the bars to start exactly from the x-axis. So again, I'm gonna put it in another layer. Say scale underscore y continuous. Inside this, I'm gonna give limit equals, all right, let's forget about limits for once. Expand equals, and I'm gonna we have to give in a list, so x and y values. So I want to start it from 0 0. So c 0 0, and it is touching the x-axis. That's what I wanted. Now, while we try to put the significance values and the significance lines, we need a space. We need a little bit space above this. So for that, I'm gonna use the limits function. Again, I have to put in the minimum and the maximum limit. So as a list. So from 0 to 3, so it will go up to 3. Maybe we can change it later. Right, for now let's keep it till here. And next, what I want to do, I want to show the data points on top of the bars. So all right, let's bring it back to 2. Back to 2. It's not happening. Okay, remove the plus. What is wrong? Select this, enter. All right. So right now we have till 2. Now I want to show the data points. Now geom underscore jitter. So this will give you the data point. So we call it a jitter plot. Inside geom jitter, again, we have to specify the aesthetics. X equals treatment and y equals fold change. Now if we give if we put mean FC over here, if we put mean FC over here, mean FC is just one value, right? So it won't have 3 different values. So for that, we are going to take the values from here, okay, from the chunk of data that we did while we were doing the data cleaning. All right, so we're going to take the value from here. So we're going to take ATP data. So in that we have fold underscore change as our column where we have the values. And if we put it like this, what will happen? It will throw us an error. Okay, So object fold change could not be found because it is not inside this ATP. All right. I mean, it's inside this ATP, but it is while we piped this, we had already made the mean of that fold change, right? So in order to bypass that, we need to specify the data set from where we are getting the fold change. Okay, if we do this, it should work. You have the data points jittered around the bars. Now you can see some is totally on the right, some is in the middle, some is on the left. I want to have a specific width in which it should jitter. So width is equals, let's give it 0.2. Yeah, this looks fine. Width is 0.2 and I want to change the alpha level to 0.5. Okay, not 0.5, it's too light. 0.7. Yeah, that's better. Color is already black by default, I like that. And finally, another thing that I want to do is change the size of the plots. So size equals 0. Size equals, okay, size does not go with decimal values, it should be like full numeric values. So size equals 3, and yeah, I like it. If I give 2, it is also good. I like 3. Let's go with 3. All right, so we have the data points and we have the bar graphs, the error bars. The last thing that remains is putting the significance values. So for that, I need the values. But before that, I'm gonna put the lines on top, just lines. So one line is going to be here till here, and compare control 40 millimolar glucose, 40 millimolar glucose to the last group GLC plus LXA4. Finally, control with LXA4. So I'm gonna add another layer of the significance lines. So this we are going to get from geom_signif. That is why we installed this package, ggsignif. This is going to give us all the tools that we require for putting in the significance lines and the significance values. First thing that we need to specify is the comparison list. So with what we are comparing what. So comparisons equals, and put in a list, like we need to put 3 lists actually. So the first is going to be glucose, first is going to be control, comma, 40 millimoles of glucose. All right, so 40 millimoles of glucose. So we need to write the \n because it's going to take the if you do not give this, it's not going to recognize it. Okay, because when we mutated it, when we recoded it, we had put this \n, and this is now a part of that name. All right, so we need to write list over here. To write list over here. Yes, so all right, I'm gonna write this again. These brackets are confusing. List C control 40 millimoles glucose. Next again C, I'll just copy this. Right, GLC plus LXA4, and the final one, it's going to be C control comma L 100 nanogram per ml. Yeah, that's it, I guess. And if I hit enter, nanogram per ml. All right, so be careful with the brackets because there are a lot of brackets going on. So you need to calculate the number of opening brackets and the closing brackets. That should be same finally. So now we have the significance lines, but all of them are in one line. So we need to adjust those, and all of them are having one. So we need to adjust that as well. Okay, so comparisons, it's done. Next, we will set the significance map to true. Map significance levels, this should be equal to true. Now if I hit enter, it's going to be non-significant for all the groups, right? And finally I can set the y positions of each of the, so y underscore position equals, over here I can put in a list of values for each of the significance lines. So it should go in this order. The first line should be for control the first comparison, the second line for second comparison, the third line for third comparison. So there we put let the first one be at 1.2, the second one be at 1.6, and the third one at 2. Put this. Okay, so the one that is at 2, it got cut from the picture. So what we can do, we can increase this. We can do 2.5. Now you can see the last one. So after this, we need to change all of this ns into the specific values. For that we need annotations equals, and we're going to write the specific values. So what I'll do, I'm gonna run this once more so that I can get the values in the console when I write it. So over here, annotations, again we have to put in a list. So first value, that is control and 40 millimoles of glucose, is this one. P equals, I'm going to just copy this and paste it over here, and I'm going to show the star value as well. So \n width, so this is 2 star, right? Okay, the next one is 40 and glucose plus lipoxin, 40 glucose plus lipoxin. The second last one, this one, it's not significant, so I'm gonna get P equals this value \n say not significant, we'll write ms. Third one is control and only lipoxin. So this one p equals this value max slash n significant. If we do this, it should be fine. Yeah, right. Now we got the star values and the p values along with the stars, and also we have the significance line. One last thing that I want to do is increase the font of the x and the y axis labels. So for that, no, not for the x and the y axis labels, for the significance value labels. So for that I can write, see there, and text size is equals, let's say 4. Let's see. Yeah, that's good. That works for me. And yeah, that's it. Now you have a pretty good graph, and you have the significance levels, you have the data points, and you have the star values, the significance lines, you have various different components, the x-axis and the y-axis, the treatment, and also a title on the top. Now if you want to export this, if you want to save this as an image, all you need to do is go to export and save as image. So over here you can specify the height and the width, and you can specify the format, and you can specify the file name. So we have PNG, JPEG, TIFF. Normally we do in TIFF. So you can do that and you can hit save, and it will be saved in the specified folder. So that's about it. So you learned how to manipulate data, how to clean data, how to do statistical analysis, and how to draw a publication ready graph with R programming.